Professor Hawken here. And that announcement is this: 85% correct. That is your average on the first exam. So I was really happy with that. Give yourselves a hand. Very nice. So I think everybody as a whole did really well. And for those of you that maybe didn't get as high a score, the really good news is that this is only the first exam. You have another midterm, a final, all your homework. And that's what your TAs are here for, to make sure that you get back on track, and maybe you weren't studying for the exam in the right way. Go see your TAs now, and we will make sure you're on the right track for the next exam, so everybody can be up at those high scores. So as I said last week, we have a very special treat for the next 2 lectures. Professor Hawken is the professor of brain and behavior in the spring semester, and we've done a little lecture switching. And it's always a treat to be able to talk about your own area of research. And Professor Hawken is an expert in visual perception and the visual system. So we're in for a treat in the next 2 lectures, where he will be giving us our 2 main lectures on the visual system and visual perception.
Okay, does that sound good at the back? And the folks who are listening, that's all right too. Okay, so Wendy graciously agreed to do 2 lectures for me in the spring semester, because you can probably tell from my accent that I'm not actually an American. Where do you think I'm from? We just beat Australia in the semi-final of the rugby. Yes. Sounds like Australia, right? So anyway, my relatives are having a big reunion, and I'm going back to visit them for a week, and Wendy's going to do my lectures in November. No, not in November, in February. So what I wanted to do was to take the rubric that Wendy had given you for the general sensory systems and specifically adopt that to the visual system. So I took her slide, which you can see up here, which gives 5 main stages of how sensory systems use information from the outside world, bring it through sensory transduction, encode that information into neural signals, send them across pathways. And one of the things that we'll learn about sensory systems is that what's out there is taken in and it's completely reinterpreted by the brain. So what you see, what you hear, what you feel, what you taste has only got a small amount to do with what's out there. It's how your brain actually interprets it. So we'll see that you need to have particular neural apparatus to be able to perceive objects, colors, depth. And if you don't have that apparatus, you don't see them. It's as if, for you, they don't exist. So I like to say that right at the beginning, so that although it seems as if there's a lot of neural work going on, you want to think of this as being what you are seeing at the moment, at the time that you're seeing it. And I can summarize some of these things by, And I didn't even look at my screen, sorry. And you didn't call out. That's better. Okay, so you can look at some of these things by saying, okay, we have transduction, sensory encoding, and then together, the neural pathways, the receptive fields, and the topographic maps come together to allow you to represent the image, the visual image, and from that image, you're recognizing objects, you're seeing things move, and you're remembering things from your previous experience, and you're comparing them with what you're seeing. But not everything that you see is exactly as it is out there in the world. And so this is very nicely illustrated by one of the first figures from your chapter 10 of your textbook from Ted Adelson's work, where he shows just a strip of vertical grey levels running from dark to bright. And you can see them, that they look darker on this side and brighter on that side. That's very obvious. Now, if you do something, you embed the same grey level, these two are actually the same grey levels, with different surroundings. They look different to you. They look different to me. Do they look different likenesses to you? Yeah, everybody sees them as different likenesses. And you can see part of the reason is that this guy is surrounded by bright, and so he looks darker. These ones are surrounded by different colors on a different surface that you see. You're also interpreting the light as coming down from here, so this is shadowed. And so all of these things, the context of something actually changes the way that your brain interprets it. And so what we want to do is to understand something about how these different brightness levels are encoded. But the other thing that we do is that we see things not just as, let's just go back here for 1 second, not just as brightness levels, but we see changes in brightness, contrast, and that gives us what we think of as edges. And if you just look around you now, almost everything is defined by an edge. You know, people's clothes are striped, they're dark and they're light. You don't really notice the light part or the dark part, you notice the edge. You notice the contrast, the difference between the brightness and the darkness. And this is something that all sensory systems are very good and adapted to actually signal to the brain. You're interested in contrast, whether it's a change in something that's moving, a difference in color, something that's contrasting in different depths, for example, in the visual system. So how do we get this information that's out there into the brain? Well, as in all sensory systems, the first thing that we do is we transduce it. And transduction, as you remember, the first point on the slide of 5 points was that you take a physical stimulus energy and convert it into a neural signal. So you have to take the light energy from out there in the visual system and convert it into something that your brain encodes. We want to encode a range of intensities. So remember the last slide with the different brightnesses and darknesses. We want to encode each of those as being different, because that's what we can perceive. And we want to, as I said, respond to changes in intensity, because that's what we see. We see edges. And so that's what transduction is. And the rest of your brain is about producing signals in terms of the local changes in voltage in membranes and the action potential codes across these different neural pathways. And that's what we'll call central processing. So as we said, transduction is the detection of the outside energy, so that's the light coming into the eye. And in terms of vision, it's transducing the different wavelengths of visible light. And that's what we call the light energy, because you know that the electromagnetic spectrum runs from very short wavelengths, X-rays and such like, through visible light to long wavelengths, radio waves. And we only detect a very small part of that. And then, as I said, what the visual system wants to do is it wants to encode a range of intensities. So what are intensities? What is the light that's coming into your eye? It's a bunch of photons. And you can think of the intensity as being related to the number of photons falling on a small area, let's say a millimeter squared, in a second. So if you have a very dim light, it'll be very few photons. A very bright light, it'll be many millions and billions of photons. And then I said what the system wants to do, it wants to signal changes in intensity. And so what you want to do is signal the relative change in the number of those photons. So how does your eye do it? Well, you all know from high school biology, of course, that light comes into the eye, shown here, and it enters through the front of the eye, going through the cornea and the lens to the back of the eye where the retina is lying. So this is a piece of retina that's taken out and laid as if you're looking at a section through it there. And we can see in the next slide that it's slightly bigger. And one interesting thing that you may or may not know is that the retina is lying with the ganglion cells, and we'll learn what those are soon, facing the incoming rays of light. So the light actually has to go through the layers of the retina to the rods and the cones, which are the photosensitive elements of the eye. So light, these photons are coming in. You can imagine a whole bunch of photons coming in, and you have to have some way of transducing those, and that's done by these receptor cells, and you all know that they're in your eye, you have rods and cones. But first of all, let's just think a little bit about what you want to have for the stimulus itself. So the stimulus is the image. So if you're looking at me or you're looking at this picture, what you want to do is have that image perfectly, beautifully focused on the back of your eye. And, if you're like me and you take your glasses off, and, I can see you guys out the back quite well, but things that are close up are quite blurred. And other people will take their glasses off and have distance blurred and close-up things in focus. And as you get older and older and older, you just have one thing in focus and you can't see anything that's close to you or anything that's far away. So, a treat that's going to happen to all of you. So, how do these images actually get beautifully focused on the back of the eye? Well, as you know, if you have an image and you have to focus it on something that's not so far away, you need to have a lens which brings those rays, which normally are coming in here and would focus somewhere way over here. You have to bring them and converge them to a point which is close by. And so that's what happens with first of all, the cornea. actually that's the strongest lens of the eye. And then again, through the lens of the eye. And the process of actually changing an image from something that's far away to being in focus from something that's close up is called accommodation. So, and the accommodation is done by the lens of the eye. So when that lens is very flat, normally objects that are far away will be in focus on the back of the eye. And then when you look at something very close, so if you put your hand right up close to your face, and in fact you can all do this, if you bring your hand and bring it in closer and you know, look at something, look at the ridges on your hand and bring it in closer and closer and closer till you get to a point where you can't see it. Yep. So that's testing the limit of your accommodation. What your lens is doing is it's getting fatter and fatter and fatter till it can't get any fatter. Now, if you're a baby, and you often see babies, they can see things right up here because their lens, as you get older, your lens gets stiffer and stiffer because the proteins actually are not as elastic, and you have less accommodation. So a baby has, 20 diopters of accommodation. You probably have, depending on how old you are, 14 diopters of accommodation. Wendy, who's 39, has probably, 7 or 8 diopters of accommodation, no, maybe a little less, and I only have 1 or 2, right? And that's why I wear continuous glasses, so I can see things out there, and I can be in focus here because I'm looking through the lenses that are at the bottom of my eye. So that's what accommodation does. And so as you get older, the lens gets stiffer and stiffer, and you get presbyopic, and that's why you need to actually have glasses on. So the other thing that the eye has is a pupil, which controls the amount of light coming into the eye. So there's a lot of light, the pupil constricts and reduces the amount of light. If you go into the dark, the pupil expands and lets more light in because you actually require that to be able to see. And the other thing that's not shown here, but it's really central to eyes and where they look, are the extraocular eye muscles. So in each of your eyes, you have 6 eye muscles, and they move your eyes around. And normally we make, a bunch of different kinds of eye movements, but 2 of the main sorts that are voluntary are called, one's called saccades, and they're the ones that if you're looking here and now you rapidly move to here and rapidly move to here, they just take your eye in these jumps. And those are saccadic eye movements. And they're controlled by vision because you want to keep things in the center of gaze where you have the highest acuity. The other thing that eye movements do is that if you are looking at my pointer, you'll be using a different kind of eye movement. You'll be tracking it smoothly. And those are called pursuit eye movements. And humans and primates actually have the best quality pursuit eye movements. Many other animals, for example, rodents, don't even make very good saccades. They mainly move their heads around. A cat, for example, will make lots of saccadic eye movements, but doesn't have the neural apparatus to make smooth pursuit eye movements, i.e. follow things. So, the nice thing is that you could see an aeroplane in the sky and you can follow it. If you have a cat, it'll be making an eye movement which will be jumping from one place to another. And so, as I said to you at the beginning, a lot of what you can do depends on what your brain has in place. So, just a little diversion into getting a good image and what the other parts of the eye do. But we want to get this image and we want to transduce it. We want to turn it from the physical wavelengths and the photons into a neural signal that we can understand. And that's done by the photoreceptor cells at the back of the eye, the rods and the cones, so called obviously because of their shape. The rods have a segment which is called the outer segment, this part of the cell. So this is a whole cell with this unusual shape. This is the inner segment. This is where the synapse is made with the next cells in the retina, called the bipolar cells. And this is the cone cell. It has a different shape of outer segment. But each of these outer segments has stacks of what are called discs, and those discs are filled with a photochemical called the photopigment, which is shown here in cross-section as stacks. And that's the chemical that is sensitive to the photons of light. So what we want to do is understand how transduction works, how those photochemicals turn the light, which is coming in this direction, into an electrical signal. And so you've all learned about how cells have a potential across their membrane, and that you can record that potential across their membranes with a little tiny microelectrode that you can push through the cell membrane, as is shown here, and you can connect it to an amplifier, and you can record the electrical potential between the inside and the outside of the cell. So that's what transduction is in all sensory systems. It's taking the physical energy from the outside world and turning that into a change in electrical potential. And we call that the local potential or the receptor potential. And that is what is shown here. So we've got the little tiny microelectrode stuck inside the cell, and what we're doing is recording the electrical potential between the inside and the outside of the cell. So do you remember what the potential of most neurons would be if you put an electrode inside them in your brain? minus 60, minus 70, something like that. If you were a muscle cell, might be minus 85. Depends how much what is leaking across the membrane. What causes this potential? Sodium, but if it's a resting state, what's mainly causing it? Potassium, excellent. You got it. What is strange about this photoreceptor? Where's its resting level? Yeah, minus 35. It's way away from the minus 60 or minus 70. And now we give a flash of light, and you'd sort of expect, you want to detect something, so you'll be at a resting level, and normally if a synapse is working and it's excitatory, like it releases glutamate or acetylcholine, the cell usually depolarizes and gets less negative. Yep. And then you reach the threshold for an action potential and you fire an action potential. That's what we've learned. But these photoreceptors, the photoreceptors in your eyes, are sitting at a negative potential of about 35. Along comes a flash of light. Sometime later, we're recording with our electrode, you get a increase in polarization, a hyperpolarization. The photoreceptors hyperpolarize to light. And we'll see how that actually turns into, at the output of the retina, actually a bunch of action potentials. So this is an interesting departure from what you probably have learned so far about most neurons. But the totally cool thing about these photoreceptors that's shown in this slide is that if you give them a dim light, they'll give a small hyperpolarization. Give them a medium intense light, a bigger one, and a bright light, a bigger one. Their responses, their amplitude of the responses are graded with intensity. And that's one of the central things that you see in all sensory systems. So, did you do touch? Yeah. So you did touch, and you saw that if you were recording the action potentials from a receptor in your finger, which responds to light pressure, that as you change the pressure from being very light to medium to intense, that at the receptor level, you got a depolarization that was graded with the intensity of the indentation. So this is exactly the same. And so one of the things that you want to think about to put all these things in perspective is that the brain uses all these same principles for many of its systems. So sensory systems generally transduce things and have graded responses of amplitude for their potentials. So how do we get this to happen? So we have some light and we turn it into electrical potential. What's shown here is the blow up of one of those discs in the outer segment of a rod, and it's stacked with chemicals. And we're not going to ask you to remember every step of this change from where light is entering and activating the rhodopsin, which is actually made up of retinal, which is actually just vitamin A, and an opsin protein with a whole lot of subunits attached. But the critical thing for you to think about here is that this photon of light comes in and it causes a set of chemical changes called a cascade. So one leads to another, leads to another. And the general principle of being able to understand this cascade is that you know from all your work up to now that the membrane potential of the cells, that resting voltage level, is determined by ion channels, by the potassium channels and by the sodium channels, causing the resting potential and the action potential. So, photoreceptors have sodium channels that are either open or closed depending on what chemical gradients there are within the cytoplasm surrounding the discs of the photoreceptors. And the critical thing that happens, and I'll just say it, but you don't need to learn it, is that light comes in, it actually uses a protein which changes the amount of this phosphodiesterase, which hydrolyzes a level of cyclic GMP inside the cell, and that cyclic GMP was bound to the receptor and it was keeping it open. And so the light comes along, it changes this enzyme, that enzyme turns the cyclic GMP into a different form of GMP, 5', and the channel closes. So one photon of light can actually lead to the hydrolyzation of thousands of these GMP molecules, the closing of thousands of channels. And if you close the sodium channel, normally the sodium depolarizes the cell. Close the channel, it hyperpolarizes. So now you want to go home and tell your mum or your dad that you know how transduction works in the photoreceptors of the eye. From the light through the chemical cascade to the opening and closing of the channels to, as we close the channels, we reduce the amount of sodium, cell is less depolarized. So all those principles you learned before about, the amount of sodium and potassium inside the cell and how the cell is hyperpolarized and depolarized, they all come into actual play and meaningful sense here when you consider what they're doing to the photoreceptor. And you know a lot about transduction, actually. So the photopigments lead to cell hyperpolarization, and the light is transduced by the rods and the cones. But we only gave you an example from one actual set of photoreceptors. We talked about the rods. The cones work in the same way. There are some things about rods and cones, however, that are very different. And one of these is what leads us to understand that we all know is between vision at night and vision for humans during the day. So when you go out at night, everything is kind of grey and dim, and if you walk out from a bright room to a dark room or a dark environment, it takes a while to adapt. We can understand all these things by knowing about how the rods and the cones respond to different intensities of light. I already told you this, but I'll just remind you that the photons that the rods and cones are transducing come from a very narrow spectrum of the overall spectral system that's out there, from very short wavelengths to very long wavelengths. And if we take that set of light and we look at how bright it is on a scale where we measure the number of photons per millimeter squared per second, right? And we record that with some measurement. The intensity, we can convert that into a scale. You can see that on a logarithmic scale, it covers 10 to the minus 9 to 1, 10 to the 10. That's 10 billion range of sensitivity. So that's pretty incredible. From the lowest level of light that you and I can detect up to really dazzling bright daylight. And these pictures here depict what kind of perception you see when you're adapted to that. And you can see, as I said, that at the low levels of light, you don't see colors. So not only in day that you see colors. And that's depicted here. So I said that the rods and the cones do two different things for us. Rods are sensitive to low light levels, up to about 10 to the minus 5 on this scale. And the cones only become sensitive at higher light levels. So we have two systems to actually encode this huge range of intensities, the rods and the cones. But even so, you know that, for example, the rods can detect a change in about 10 to the 4 of intensity here, but neurons only have a range of their responses that are maybe from 1 to, 100, at the best, right? They have a dynamic range that changes much less than this range here. So how does the rod, or how do the rods and how do the cones actually allow us to see this huge range of intensities with a system which has a fairly narrow dynamic range, a fairly small dynamic range? Well, we just showed you one thing. It has rods and cones that allow you to work over two different ranges, but that's not all, because that's not taking care of something like 10 to the 2, the number of action potentials the neurons can fire. By up to 10 to the 4, you would need 100 different levels of, or different types of receptor to be able to actually encode between 100 and 10,000. And so we are able to see different kinds of objects because in different light levels, because we and squirrels have cone dominated retinas. Actually, we have a combination of cones and rods. We have more rods than cones, but rats and mice, for example, that live mainly in the dark or in low light levels and come out only when the sun is just coming up, have a rod-dominated retina. So as I told you before, what you can see depends on what neural apparatus you have. And some monkeys, like this owl monkey here, it has also a rod-dominated retina. It is mainly living in low light levels in the forest at night, and it often has a really wide pupil to let in lots of light. Remember I said that the pupil determines how much light you can see, can get in. So the two systems of receptors allow us to work at different ranges, but we also have a system in all photoreceptors that's another system. It's called adaptation. And the photoreceptors change their responses to work at whatever ambient level of light there is present in the environment. So your eye is actually changing its dynamic range, its sensitivity. So when really low light levels, the rods are sensitive to changes of light of about 100 times, of about 100 in very low light levels. But if you move up to an intermediate light level, or if you showed those rods a medium light level, their response would go up to the maximum and respond, and small changes about that medium light level at that time would not be sensed by them. They would be saturated. So over a period of time, they will then become adapted so that they will then start working to changes in light level over that range of light. And you know this perfectly well from when you walk from here to outside when it's really bright. For a while you can't see anything. And that's because your cones have adapted to work in a sort of moderate light level. And when you walk outside, they get a lot of light, and they're all responding maximally, and small changes around that new light level are not signaled by them. So this is a really incredible adaptation that biology has made to allow organisms to actually work over huge ranges of intensity, which are environmentally important for them, using a combination of different cell types and mechanisms, biophysical mechanisms inside the cells to allow them to automatically adjust their range of responsiveness. So we've transduced, and now we want to, I'll call it encode. We want to take those responses of the different photoreceptors, and as I said, mainly what we respond to is not just absolute changes in brightness and darkness, but we respond to the changes in the brightness and darkness at edges. So light and dark in different image regions, and to the changes in the light and dark over time. So when something moves, the bright and the dark regions on one part of the retina will change, and that will lead to these important parts of perception. But actually, before we get there, to object recognition, to recognizing actions and visual memory, what we do is we take the responses of the cones. It's like the pixels on a television screen. And we divide the image up into the important parts that are used by the brain to segment objects, to recognize objects that are moving. And the way that our brains seem to do this is that they have mechanisms which recognize local shapes, local colors. They have mechanisms which are sensitive to different rates of movement within the biologically relevant rates of movement. And you and I are particularly good at detecting the depth of objects. And that's purely a mechanism that comes about by having two eyes and taking the information from those two eyes and combining them in certain ways to produce a neural map of things that are in depth. Actually, that's of course not quite true, because you can cover one eye and you can tell that things are in front and behind. So we use actually quite a lot of different cues for depth. But for the really fine discrimination of depth, you know, when I'm reaching out to touch the end of the pen, and you can try that yourselves now, put your pen out there, right? Reach out and touch the end of it. You can do it effortlessly, close one eye. Try it again. It just doesn't seem right. Yes? You need two eyes to be able to encode that fine discrimination of depth. And so that's where we're going to go next. We're going to combine the idea of the neural pathways, receptive fields, and maps to actually understand the perceptually important parts that build up our internal representation of the visual image. So we've got signals from the cones, and we've got a retina, and here's the retina, here are the photoreceptors, and they pass their signals from different parts of the retina up through the bipolar cells in the retina to the retinal ganglion cells, and those retinal ganglion cells send their axons out to the thalamus and other parts of the brain via the optic nerve. And so another important principle that you are probably aware of, but I want to reiterate here because it makes it easier to think about the system, that the transfer of information, the transfer of the signals, proceeds both serially, so it goes from the rod or the cone through a bipolar to a ganglion cell, and it goes in parallel, because that's happening, the same sort of process is happening at a whole lot of different places at the same time across the retina. Do you get that? So you have a big sheet of retina. I mean, this is just one dimension, but if you were looking down on it, you have all the rods and cones spaced out here, and in any part of that, if you have the same image, each of those parts is signaling to its connected bipolars and ganglion cells some of the same information. And then you have an edge. So now you have an edge here, then these cells will be signaling some different set of information. It'll be going in parallel across these different neural circuits. So now, to understand what the processing is happening, even in the retina, and how that information gets to the brain, we want to think about what's it trying to do. So let's step it back and look at a couple of aspects of perception. And I like to use the analogy of something that you probably all understand, and you may or may not have been introduced to, is what do I hear, which comes from the auditory chapter of your textbook. And what it shows is the different frequencies of sound here on the x-axis, and how intense those sounds have to be for you to just say, I hear it. And see, this scale is actually in decibels, and a decibel scale is a logarithmic scale, and a change in 20 decibels is a factor of 10 change in intensity. So this is just about threshold. This is 10 times, 100 times, 1,000, 10,000, 100,000 times louder, right? So for a 100 hertz tone, a really low tone, and you're a human, so this is the black curve, you need to have something that is about 1,000 times louder than for a 4,000 hertz tone for you just to be able to hear it. So these are the curves for just being able to, and this is the equivalent of an audiogram. So when you go to get your hearing tested, they play different tones and they change the intensity. They say, can you just hear it? And this is what you measure. So this would be called the psychophysical auditory tuning curve. And we can say, well, how does the brain represent this? What does it have neurons that respond to all of these frequencies, just one neuron, and that's what I'm hearing? Or does it have neurons which are responding only to a fairly narrow range of these frequencies? And you'll learn, if you don't already know, you'll learn that you have neurons which respond, some which respond to low, some which respond to intermediate, some to the optimal frequency, some to high. So you have a range of tuning curves. And so I just want to introduce this because what we'll see is in vision, you actually can do the same kinds of measurements. You can measure people's psychophysical ability to see objects in the world, and that's where this idea of spatial frequency comes in. But I just wanted to show you a couple of nice examples of things that tell us that what you see is not what's out there, but what your brain interprets. So, and some of the nice things come from illusions. So in the Muller-Lyer illusion, you are supposed to judge whether this length of this line segment is the same as that one. And to most people, this line segment looks shorter than that one. You're supposed to judge whether this circle embedded in the surrounding circles looks the same size as this circle, and this one, to me, looks bigger than that one. It probably does to you. And similarly, you can work out what is happening in these other illusions. One of my favorites is this one, now you want to judge whether this object is the same size as that object. Is it smaller or larger? Smaller or large? Smaller than the yellow one? Perceptually smaller than the yellow one, rather than objectively smaller than the yellow one? Perceptually smaller. It just appears smaller. There's nothing you can do about it, right? A fantastic example of this is when you go out into Broadway, look down the street towards Canal Street, and then if you look at people going across, 3rd versus Bleecker, and just look to see what sizes they appear. Just look at the people. They will appear to be the same size. You'll say, oh yeah, they're the same size. But on your retina, the people that are closer are much bigger objects than the people who are further away. So what your brain is doing is recalculating the size of objects depending on where they appear to be in the environment, right? So this object here, which is the same size as this one here physically, seems to be, because it's further away in the perspective, seems to be bigger. So we do all sorts of complicated things in vision called constancies. The other thing that you can do when you look at the people walking across the road and you're looking down the street, see whether you think the ones closer to you are walking faster or slower than the ones who are going across. So you have some people who are close to you here and they're walking across the street, other ones 1 block away and walking across the street. And then think about how fast those people are moving, or the images of those people are moving on your retina. So the people who are moving at the same speed on your retina, same speed physically, the ones who are closer will be moving across your retina much faster than those people who are further away, just because of perspective. And what you judge them to be is moving at the same speed. So we recalculate all the things in the image, sizes, depths, speeds, by the perspective. We don't actually know how the brain does this yet, but this is one of the things that we understand that makes it different from just performing these calculations on the image itself. So as I said, if you wanted to see how people hear, you can take tones of different frequencies and you can say, how well can I hear them? You change their intensity. So in your textbook, there's a whole section on these spatial sinusoids or spatial frequencies. You can think of them just like tones. So the tones are vibrations of the air going backwards and forwards in a particular rate or frequency. Now you can take a spatial image, put it on a television screen, and you can vary the brightness from here, so it's bright, dark, bright, and you can do it either as a square wave or a sinusoidal modulation. And the beautiful thing about this is that you can use this to actually test the visual system in the same way as you can test the auditory system. So you can take a certain size and you can make a number of cycles of light and dark in this. It's called the spatial frequency, and you can make 10 cycles in one particular distance. And if you actually do that for a degree of visual space, and a degree of visual space is really easy to know about, because if you hold out your thumb, hold out your hand, put up your thumb, and look at your nail, that's about 1 degree of visual space. So now, if you paint these gratings on your thumb, and you can say, how many of those light and dark bars can I see before they just become grey, a grey blur? That would be your limit of how fine a detail you can see, or your visual acuity. We normally go to the eye doctor to measure the visual acuity, but we might want to measure the visual acuity of other animals. Hard to go to the eye doctor and ask them what letters that they're going to read on the eye chart, right? So we can use these kinds of techniques, the same as you use for hearing, and actually you may or may not have learned about it, but the same as you can use for touch, by changing the frequency of the stimulus and recording the response. And so here's a grating of the same spatial frequency, and what we've done is reduce the amplitude. And the amplitude just means making the bright and the dark bars less bright and less dark. And we could just reduce that amplitude until you could only just see it. And that will be the threshold. And we could do that same thing for these gratings of different spatial frequencies. And one of the things that people have used is that they've used this idea that images are made up of combinations of these high spatial frequencies and low spatial frequencies to understand the components of images. And that's what's shown on the bottom of this graph here. So here are the pair of penguins, and what's happened is that there's been an algorithm that's been used to take out the high spatial frequencies. And what you can see is at the edges, and you can imagine that's the very small changes in local spatial scale of the bright and the dark bars become blurred. You can't see them so easily. But you can see the general outline. If you filter out the low spatial frequencies, the very slowly changing bright and dark parts in this image, you end up with just the outline of the penguin. So you miss out on understanding whether this is really dark or really light. But what you can do is you can see the outline. And you can recognize objects either filtered by low spatial frequencies or high spatial frequencies, because you actually lose some of the information when you take out both of those. And it turns out that we have channels. Part of what our brain is doing is it's encoding the low spatial frequencies, the intermediate spatial frequencies, and the high spatial frequencies by different sets of neurons, just like I told you for the auditory system. And we can actually measure that in the same way as we measured the auditory system spatial frequency tuning. So you can go from low spatial frequencies, so this is one of those gratings where the sinusoidal modulation is very broad. Like if you put out your thumb, it'll be one bright and one dark region on your thumb. That will be 1 cycle per degree. If you painted 10 of those on your thumb and look straight ahead, that will be 10 cycles per degree. And this is about 60 cycles per degree. And this measurement here is changing the amplitude of that sinusoid so that you can say, I just see it. It's just like changing the loudness of the frequency of the tone till you can say, I just hear it. So this is the spatial contrast sensitivity function. And so the nice thing about this is it allows you to determine what the sensitivity of the visual system is to the spatial components of the image, just like it allows you, the auditory function allows you to determine the auditory sensitivity. But normally we would go to the eye doctor and we would be asked to read a eye chart, and that's shown here, the normal Snellen eye chart, and you read down here till you get to, depending on what distance you are, and if you're about 6'6" in vision, that's equivalent to being able to see about 20 or 30 cycles per degree. That's about the normal human ability to see objects. There are other eye charts that take the same size objects and measure and change their contrast, so you can actually measure the contrast sensitivity of people. But it's hard to do that with animals. So what allows us to actually see this really fine detail? One of the things that we have that's a specialized region in the center of our gaze, which is called the fovea, is we only have cones and no rods. So what you already know is that when you go out at night, you can't see fine detail. So if you paint 10 cycles of a grating on your finger and go out in dark night and look at it, it'll just look like grey. You won't be able to see it. And that's because the cones are not operating, only the rods. So the detail that the rods can tell you about is really poor. The other thing that you may have already noticed is that if you look straight at your thumb, your rods won't be actually working at all in the region of your thumb because you don't have any in your foveal representation. So you'd actually need to look here and hold your thumb out at about 10 degrees to actually have the best ability to see with your rods at night. And so that's another specialization. We humans actually sacrifice the center of gaze of our retina to be able to see this very fine detail because presumably ecologically that's important to us. The other thing that you'll notice is that I told you that light actually comes through the ganglion cells to hit the photoreceptors. In the fovea, the organization is such that these parts of the circuit are bent off to the side. So the light which actually comes down here is not filtered by the neurons at all. So that's called the foveal pit. So we're specialized in a couple of ways to actually be able to see fine detail. But the other main thing about being able to see that really fine detail is that it only happens in the center of gaze. And the reason for that is because you have many more cones in the foveal representation than you do as you go more peripherally. And so that's shown here. This is the foveal representation of the eye. This is where the optic nerve goes through. These red things are the blood vessels, so you'll be looking into the eye. If you took the retina out, you took it flat, and then you looked down on it, and you looked at the inner segments of the photoreceptors. So that's what's shown in these pictures. You're taking a microscope. It has a particular characteristic that allows you to optically dissect through the layers of the retina, and you just see these inner segments. And you can see these guys in the central retina are really closely packed. And as you move out, even along the horizontal meridian here, the cones shown by red are much bigger, and there are many fewer of them. And the little blue guys are the rods. And the rods are more frequent, and they're smaller, and as you go out even further, the cones become even less dense, the rods slightly bigger, and many more of them. So the density of cones is huge in the retina, in the central retina, and falls off really rapidly as you move out to the peripheral retina, and the density of rods goes from 0 in the foveal retina up to a maximum of about 10 to 20 degrees. So that explains why we can see detail at night best away from the center of gaze, but not very good detail. And you might say, well, why can't we see very good detail? There are many rods here. The thing about that, as we'll learn next, is that's part of the serial processing of the images, right? So those rods, they become connected to one ganglion cell. So they're summing the information over a big area. And if they're summing that information over a big area, they can't tell the difference between something within that large space that they're summing over, if they're connected to a single output cell. So now you can measure how well you can see over the central retina, and this is the acuity from 100 out to 20 degrees. So if you're looking straight ahead and you have an angle of 20 degrees, you're something like only 5 or 10% of as good. So that means a grating of 60 cycles per degree here. You would have to have one which is probably 2 cycles per degree out here. So if you have your thumb out here, you can actually only just see your thumb and might be able to see 1 or 2 bright and dark bars written on your thumb. So what allows us to see fine detail is the high density of the photoreceptors and the fact that the photoreceptors are connected in a one-to-one way through the bipolar cells to the retinal ganglion cells. So it's as if there's a labeled line going from each photoreceptor through the bipolar to the retinal ganglion cell, and that labeled line from that single cone is actually going out to the brain. And there's a very nice relationship between the density of retinal ganglion cells connected to the photoreceptors and the visual acuity of different species of animals. So rats, for example, can see at best about 1 cycle per degree. So they're about 1/60th as good as you and I. A cat can see somewhere between 5 and 7 cycles per degree. The raptors, who are hunting, flying along, looking for small animals scurrying around on the ground, they can see up to 100 cycles per degree. And part of the reason for these specializations is that the hawks, for example, have really big eyes. They have really small photoreceptors that are packed into their fovea, and actually they have some optical specializations that make the light actually even more focused than in our eyes. So now we take these signals from the rods and the cones and we process them in parallel across the retina. And we can, if we understand how one of these serial pathways work, we can apply it to all of the other pathways. So we can nicely understand the 1,000,000+ cones in our retina, how they work, just by studying 1 or 2 of them. And so the signals are taken by the cone photoreceptors, and you get a hyperpolarization, which is either connected to the next cell in the retina, which is called the bipolar cell. And these come in 2 categories. One is called the off-center bipolar cell, and the other is called the on-center bipolar cell. And they respond differently to different changes in light. So remember, you could have an ambient light level, and then you could increase the amount of light. And if you increase the amount of light over the receptor, so you would illuminate the light, then this on-center bipolar cell will become depolarized, will release more transmitter and depolarize the retinal ganglion cell, which will then increase its firing rate to the increase in light. But there's another type which, if you increase the illumination to the photoreceptor, this off-center bipolar cell will become hyperpolarized, and it'll be connected to an off-center ganglion cell, and its firing rate will be decreased. Now you can imagine the converse. You could have a constant level of light, and then you could decrease their level of light, you could reduce the center illumination, and so the opposite would happen. The on-center ganglion cell will decrease its firing rate, and the off-center bipolar and ganglion cells will increase their firing rate. So now we have two systems of on and off which are actually working in parallel. And that's where I was going to actually show you the animation of the receptive fields, which wasn't working, but since it's perfect timing, I can begin Wednesday's lecture with the animation of the receptive fields for the on and off ganglion cells, and then lead into the visual pathways and get to the cortical processing.